it over to Vance, who is going to introduce today's speaker. Take it away, Vance.
Thanks, everyone. I'm Vance Friedenberg. I was a student here ages ago, and now I'm a professor at San Francisco State. And I'm very happy to introduce Dr. Javier Cuervo, who comes to us from the Museum of Natural Sciences in Madrid, where he's a researcher. He's an ecologist who's worked, a behavioral ecologist and general ecologist who's worked mostly on birds and on lizards, which we'll be talking about today. He did his PhD work in arid zones in southern Spain, and he grew up in the Rioja region of Spain, near Basque Country. So, so I'm very happy and pleased to tell you he's going to talk to us today about the function of red coloration in the spiny-footed lizard. And if anybody has questions, please come talk to him. He's on sabbatical here at SF State, and he's working in collections here, and he'll be here through the end of May. End of June. End of June. So great. Thank you very much.
Thank you, Vance. And well, thank you very much to the organizers of the lunch seminars for inviting me to give a talk. I will begin with a brief introduction about animal coloration. Colors can be interpreted as electromagnetic wavelengths that are visible by animals. In the case of humans, visible wavelengths range between 400 and 700 nanometers. But this range of wavelengths can be different in other species. For example, birds and reptiles can see in the ultraviolet. Animal colors can be classified in different types according to the way they are produced. For example, we have bioluminescence, in which light is the result of a chemical reaction. It is relatively rare in terrestrial environments, but more common in marine animals. Here I show two examples, the firefly and the lanternfish. Structural colors are those produced by the physical interaction between light and the nanostructure of the integumentary tissues. Most iridescent or blue colors in animals are produced by this mechanism. For example, the iridescent green of this butterfly, or the blue wing patch of the Eurasian jay. There are also pigmentary colors, which are produced by some molecules called pigments that interact with light and absorb certain wavelengths. There are many different types of pigments, but the most common groups of pigments in animals are melanins, carotenoids, and pterins. Melanins produce black, gray, or brown colors, for example, the black coloration in this panther. Carotenoids produce red, orange, or yellow colors, for example, the red big spot in this penguin. And pterins produce similar colors to the carotenoids, for example, the red eyes of the fruit fly. Sometimes coloration is the result of combining both structural and pigmentary mechanisms. For example, in the skin of this frog, or this chameleon, there are yellow pigments, but there are also nanostructures producing blue coloration. And the result of combining the yellow and the blue is the green color that we can see. Coloration has many different functions in animals. For example, it may help in concealment. Crypsis is the ability of an animal to avoid being detected by other animals. It is useful as an anti-predator mechanism, but it can be also useful for predators trying to avoid being detected by their prey. The flounder or the polar bear are examples of cryptic coloration. Coloration can be also useful for thermoregulation, which is the ability of an organism to keep its body temperature within certain boundaries. Of course, the effect of coloration on body temperature is more important in ectoderms. For example, the melanic morph of the common garter snake is able to maintain higher body temperatures than the striped morph. Coloration can be important in intraspecific communication in many different contexts, for example, parent-offspring communication, sexual selection, or the more general social selection. Here I show some examples. Mouth coloration in band swallow nestlings is used by parents to allocate food. And orange spots make male guppies more sexually attractive to females. And blue and red colors in male mandrills indicate dominance, both in sexual and non-sexual interactions. Coloration can be also important in interspecific communication. In this case, signals are often directed to potential predators. For example, the aposematic coloration of the ladybird is a warning that this animal is toxic. The black spot on the tail of this tadpole is a decoy for diverting predator attacks from vital body parts, for example, the head. And tail flicking in the moorhen shows the white undertail, and it signals to the potential predator that it has been spotted and an attack will probably not succeed. After this introduction, I will talk about the spiny-footed lizard, the species I've been studying during the last years. It is one of the around 40 species of the genus Acanthodactylus within the family Lacertidae. Total length is up to 23 centimeters. Males are generally larger than females. And one characteristic of this and other species of lizards is tail autotomy, because the tail breaks when the predator grasps the tail, so allowing the lizard to escape. The species is distributed in the Iberian Peninsula and Northern Africa, and in this map I show also the location of the two populations where I've been studying the species. One of them in Madrid in central Spain, and the other in Almeria in southern Spain. Spiny-footed lizards feed mainly on insects, for example ants, beetles or true bugs. But they can eat many other small invertebrates, for example spiders, and sometimes they can eat plant material. Their main predators are snakes, for example the southern smooth snake, and birds, for example kestrels or shrikes. And even hoopoes can sometimes catch one of these lizards. The phenology of the species differs slightly among populations, but in southern Spain, the eggs hatch in August and September, and hatchlings are active until winter hibernation. Then they are active again in early spring, and during this season, they are juveniles or sub-adults. They hibernate again the following winter, and in spring they are already adults. They begin to mate in April, and females lay eggs in May or June. Coloration in this species undergoes ontogenetic changes. Hatchlings of both sexes show red coloration on the ventral part of the tail and also on the dorsal part of the tail. Juveniles of both sexes show a red coloration on the ventral part of the tail and on the rear part of the hind limbs. Males lose their red color when they are around 1 year old, and so adult males show white coloration on the ventral part of the tail and the hind limbs. In contrast, females retain the red color until they are adults. At the beginning of the breeding season, they show red coloration on the ventral part of the tail and the hind limbs, but this red color gradually fades and becomes white, yellowish white, when females are gravid. Our first question in relation to the red coloration in the spiny-footed lizard was what kind of pigment is responsible for the red color. In other lizard species, red coloration is caused by carotenoids, for example in the Australian field-neck lizard, or by pterins, for example in the striped plateau lizard. The kind of pigment responsible for the red color might be important because carotenoids in general can't be synthesized de novo by animals and need to be acquired in the diet. And pterins can be synthesized within animal cells. In any case, both types of pigments seem to be involved in oxidative processes and might play a role in immunocompetence. To answer this question, we separated carotenoids from pterins in a sample of red skin from the lizard using different solvents. Carotenoids should remain in the upper phase and pterins in the lower phase. Using absorbance spectrophotometry, we found that there were pterins but not carotenoids in the sample. Then we compared the pigment found in the lizard with Drosopterin from fruit fly eyes using thin layer chromatography and spectrophotometry, and found that both pigments were identical. So the pigment responsible for the red color in the spiny-footed lizard is Drosopterin. A second question in relation to this color was if red coloration is proportional to the amount of pigment. The answer to this question is not obvious because in reptiles and in other animal groups, for example cephalopods or amphibians, pigments are contained in special cells called chromatophores. And then the pigments may or may not be visible depending, for example, on the shape of the chromatophore or on how dispersed pigments are within the cell. In addition, tissue nanostructure, for example, the layer of iridophores, can modify pigmentary coloration dramatically. To answer this question, we measure with a spectrophotometer reflectance on the ventral part of the tail in adult males, gravid adult females, and juveniles to have the full range of coloration found in the species. With this reflectance data, we calculated brightness that tells if the color is light or dark, and red chroma that represents redness. Then we quantified pigment concentration in skin samples from the ventral part of the tail using chromatography and tested the relationship between color parameters and pigment concentration. We found that both relationships were highly significant, with darker and redder colors in samples with higher pigment concentration. The two relationships remained highly significant when only juveniles were included in the analysis. So the answer to our question is yes, red coloration in the spiny-footed lizard is proportional to the amount of pigment. Now I'm going to talk about the function of the red color in the spiny-footed lizard. This function might be different in different life stages, and in fact we had different hypotheses for hatching juveniles and adults. These hypotheses were the decoy hypothesis in hatchlings, the aggression avoidance hypothesis in juveniles, and the sexual ornament hypothesis in adult females. In the three cases, in the three hypotheses, the red color has a signaling function. I will begin with the decoy hypothesis in hatchlings. According to this hypothesis, red tail coloration diverts predator attacks from vital body parts to the expendable tail, then increasing the probability of surviving the attack. The hypothesis is based on two characteristics. The species shows a tail autotomy, and red tail coloration in hatchlings is visible from above, so it's visible to predators. As I said before, the decoy hypothesis implies a signaling function, and the receivers of the signal are visually directed predators, mainly birds. The message of the signal is something like "attack here," and this hypothesis has been previously tested with blue tails, which are common in juveniles of some lizard species, for example, in this ink. According to the decoy hypothesis, the red color has an anti-predator mechanism. Then one prediction of the hypothesis is that red-eye lizards will perform more risk-taking behaviors. To test this prediction, we made behavioral observations of the lizards in the field and calculated 5 behavioral indices that were presumably related to risk-taking behaviors. They were movements per minute, percentage of time spent moving, mean distance to refuge, tail displays per minute, and limb displays per minute. After the observations, we tried to capture as many of these lizards as possible. Then we measured snout vent length to have an estimate of body size, and also measured coloration, again using a spectrophotometer, on the ventral part of the tail and the rear part of the hind limbs, and calculated brightness, red chroma, and hue. Hue is what we usually call color, and was estimated as the wavelength with the maximum reflectance. We found that movements per minute or percentage of time moving were not significantly related to lizard coloration, but movements per minute was related to snout vent length. Mean distance to refuge was the only behavioral index significantly related to lizard coloration. Specifically, it was related to hue. Regarding the relationship between movements per minute and snout vent length, smaller individuals were more active than big ones. And when snout vent length was replaced in the model with age class, then hatchlings had a higher movement rate than adults, while juveniles had intermediate values. And regarding the relationship between mean distance to refuge and hue, we found that individuals with higher hue value, that means individuals that were redder, less orange, stayed farther from refuge for longer periods of time. That means that redder individuals were behaving more riskily, a result that is consistent with the anti-predator function of the red coloration. Another prediction of the decoy hypothesis is that red tails will make lizards more detectable to visually directed predators. To test this prediction, we made plasta and plasticine lizard models of 2 color morphs, with red or with a striped tail, based on the coloration of spiny-footed lizard hatchlings. Lizard models were placed in the field and inspected every 3 or 4 days, and predator attacks were identified by the marks left on the models. For example, this is the mark left by a bird on a plasticine model. This is the mark left by a bird on a plasta model. And these are tooth marks left by a lizard on a plasticine model. In the analysis, we only included attacks by birds because birds are mainly visually directed predators. We estimated detectability by predators, analyzing the number of days until the models were attacked. and found a significant interaction between type of model and color morph. In separate analysis of plasta and plasticine models, we found that models with red tail were attacked sooner than models with striped tail. But this only happened in plasta models, not in plasticine models. So at least for plasta models, red tail seemed to make models more detectable to predators, a result that is consistent with the decoy hypothesis. And the key prediction of the decoy hypothesis is that red tails will divert predator attacks from vital body parts. And indeed, we found that models with red tail received a larger proportion of attacks on the tail than models with striped tail. And this difference was marginally significant. To summarize the studies about the decoy hypothesis in hatchlings, we found results consistent with 3 predictions of the hypothesis, and in general they suggest that red tails are effective decoys for avian predators. However, our results were not clear-cut for a number of reasons. For example, risk-taking behaviors were not related to red chroma, only to hue. Red tails did not make plasticine models more detectable to predators, and the difference in the proportion of attacks on the tail between models with red and striped tail was only marginally significant. Now I will talk about the aggression avoidance hypothesis in juveniles. According to this hypothesis, red coloration reduces aggression from conspecific adults because it indicates subordination or sexual immaturity. The hypothesis is based on 2 observations. The species is territorial and aggressive towards conspecifics, and red coloration in juveniles is clearly visible from behind, so it's visible to other lizards. The receivers of the signal are conspecific adults, mainly males because they tend to be more aggressive, especially during the breeding season. The message of the signal is something like, don't attack me because I'm not a competitor. And this hypothesis has been previously tested in all these lizard species, but different studies reached different conclusions even within the same species. A prediction of the aggression avoidance hypothesis is that naturally red juveniles will be attacked or beaten less by conspecific adults. To test this prediction, we did a study in captivity. We measured juvenile coloration, again using a spectrophotometer, and again on the ventral part of the tail and on the rear part of the hind limbs, and calculated brightness, red chroma, and hue. Then we videotaped 30-minute encounters between one adult and one juvenile, and counted the number of attacks and bites received by the juvenile. We found that juvenile coloration was not significantly related to the number of times that juveniles were attacked or beaten by adults. So these results are not consistent with the aggression avoidance hypothesis. A second prediction of the hypothesis is that juveniles experimentally painted white will be attacked or beaten more by adults than juveniles painted red. To test this prediction, again, we did a study in captivity, and naturally red parts of juveniles were experimentally painted using either white paint, imitating the color of adult males, or using red paint, imitating juvenile coloration. Then again, we videotaped encounters between one adult and one juvenile and counted the number of attacks and bites. Juveniles painted white and juveniles painted red did not differ significantly in the number of times that they were attacked or beaten by adults. The sex of the adult was the only parameter significantly related to the number of times that juveniles were bitten, because in general males were biting more than females. However, when number of times bitten was controlled for the number of times attacked, then there was a significant difference between the two experimental treatments. This relative number of times bitten indicates the probability of an escalation in aggression intensity from attacking, that is considered low intensity aggression, to biting, that is considered high intensity aggression. And escalation in aggression intensity happened more often in juveniles painted white than in juveniles painted red, a result that is consistent with the aggression avoidance hypothesis. Then, to summarize the studies about the aggression avoidance hypothesis, our results did not support the first prediction, but were consistent with the second prediction if we consider escalation in aggression intensity. Our interpretation of these results is that red coloration in juveniles reduces aggression intensity by adults, and that result is consistent with the hypothesis. However, small variations in the expression of the color don't have any further effect. That means that the red coloration is indicating sexual immaturity or subordination, but it doesn't really matter if the red color is more or less intense. Well, in any case, the results were not clear-cut either. For example, because coloration did not affect absolute aggression levels, only escalation in aggression intensity, and because the sex of the juveniles couldn't be determined. And finally, I will talk about the sexual ornament hypothesis in adult females. According to this hypothesis, red coloration makes adult females more sexually attractive to males. The hypothesis is based on two characteristics of the red color. Red coloration in adult females is visible from behind, so it's visible to other lizards. And the red coloration disappears when females become gravid. The receivers of the signal are conspecific adult males. The message of the signal is something like "mate with me." And this hypothesis has been previously tested in other species, for example, with the orange ventral patches of females in the lake dragon lizard. One prediction of the sexual ornament hypothesis is that red coloration will increase shortly before mating. In other species, the prediction would be that the ornamental color will appear shortly before mating, but in the case of the spiny-footed lizard, the red color is already present in juveniles. To test this prediction, we did an experiment in outdoor terraria and also made field observations. Well, in fact, the reason to perform an experiment was to find out which factors were triggering a female color change. We had 3 experimental groups, one female with another female, one female together with a male, so fertilization was possible, and one female with a male, but separated by a transparent methacrylate wall with holes, so the female could see and smell the male, but fertilization was not possible. In this way, we could know if a sexual stimulus, for example, the presence of a male, or a fertilization were necessary for color change to happen. Females involved in the experiment were captured in March when reproduction had not begun, and during the time they spent in captivity, their coloration was measured 5 times. And simultaneously, we captured and measured the color of free-ranging females. Well, coloration again was measured with a spectrophotometer on the ventral part of the tail and the rear part of the hind limbs, but this time instead of calculating brightness, chroma and hue, we did a principal component analysis on spectral values to obtain the factors that explain color variation. The first factor, PC1, represented mainly brightness, and the second factor, PC2, represented mainly redness. Well, we found interesting effects of female body size on the timing of color change. But in this table, I would like to highlight two results. First, that there was a color change, both in brightness and redness. And the second, that the color change in redness was very similar in the three experimental groups. In these graphs, solid symbols represent the females in captivity in the three experimental groups, and open circles represent free-ranging females. The change in coloration is shown in the graphs by an increase in PC1 brightness and a decrease in PC2 redness. The reduction in redness was less pronounced in free-ranging females compared to the females that were in captivity. But maybe the most interesting result here is that there was a significant increase in redness at the beginning of the breeding season, a result that is consistent with our prediction. It was also interesting to find that females from the three experimental groups became gravid and laid eggs, but only females that had been able to copulate with a male laid fertile eggs. So this suggests that males have very little effect on triggering female reproduction or female color change. And there must be external abiotic factors that induce the physiological process responsible for both breeding and color change. Another prediction of the sexual ornament hypothesis, probably the key prediction, is that males will prefer to mate with redder females. To test this prediction, we did a series of experiments in captivity. We manipulated experimentally the coloration of the females using white or red paint in a similar way as we did in the experiment with juveniles, painting the ventral part of the tail and the rear part of the hind limbs. And males were offered pairs of females that differed in coloration and sometimes also differed in size and maturity, because we were using both adult and sub-adult females. Sub-adult females were juveniles, but large enough to allow us to determine their sex, and large enough to have reached the minimum size for reproduction. In each trial, females were placed in transparent plastic compartments, one on the right side and the other on the left side, separated by an opaque wall, so males could see both females, but females couldn't see each other. We considered two ways of assessing male choosiness. Time spent in contact with the stimulus. In fact, it was in contact with the transparent wall, because contact with the female was not possible, and time spent close to the stimulus, specifically in the selection areas that are shown here in gray. We did 3 experiments. In experiment 1, males were offered 2 females, one of them adult and the other sub-adult, both with their natural coloration. All sub-adult females showed red coloration, but all adult females had already lost the red color and they were white. In the second experiment, we reversed the natural coloration. Adult females were painted red and sub-adult females were painted white. Finally, in experiment 3, males were offered 2 adult females. One of them painted red and the other painted white. These are the results for experiment 1. We found that males spent more time in contact with the transparent wall of the adult females. So it seems that males prefer large white females instead of small red ones. In experiment 2, males spent more time close to adult females painted red. So males preferred large red females instead of small white ones. So in both experiments, regardless of the color of the females, males always prefer the large female, the adult female, instead of the small sub-adults. In experiment 3, males spent more time close to adult females painted red. When males were offered 2 adult females, they spent more time with the red one. So they probably prefer the red over the white female, and that is a result consistent with our prediction. To summarize the studies about the sexual ornament hypothesis in adult females, we found results consistent with 2 predictions of the hypothesis, and these results suggest that red coloration in adult females has a mating-related function. As happened with the other studies, the results were not completely clear-cut. For example, because captivity had some effect on color change, and because male preference sometimes was expressed as contact and sometimes as proximity to the stimulus. Even if red coloration makes adult females more sexually attractive to males, we still don't know what the red color is really indicating. It probably indicates sexual receptiveness, as has been suggested in other species, for example in the lake dragon lizard. But red coloration might also indicate phenotypic quality, as has been suggested in the striped plateau lizard, or even reduced Spain competition risk if the red color is related to virginity. As a final conclusion, our study suggests that the red coloration in the spiny-footed lizard is a multifunctional signal with different functions at different stages of development. We can speculate about the possible evolution of the trait, of the red color. The red color might have evolved first in hatchlings as an anti-predator mechanism. Then this color might have been associated by adults with sexual immaturity or subordination, then reducing adult aggression against juveniles retaining this color. And finally, retention of the red color by adult females as a sexual ornament might be related to the lower costs of reusing a signal instead of producing a new one. The opposite direction in the evolutionary path, with red coloration appearing first in adult females and spreading to the other stages of development, we think is less probable because males chase and bite females when looking for copulations. So we don't think it's a good strategy for juveniles to imitate the coloration of a receptive female. And to finish the talk, I would like to mention my collaborators, Josabel Bellure and Belén Fresnillo from Alcalá University, and Juanjo Negro from Doñana Biological Station. And that's all. Thank you.
That the color evolved in crotophytid lizards is the reverse sequence of the one you just presented, where in the common ancestor, adults have gravid coloration, adult females, and in crotophytus they've evolved the subadult males acquiring the color that the gravid females also have.
In which species? In which?
Well, crotophytus and Gambelia. Yeah, so in Gambelia and in crotophytus, females have gravid coloration, but just in crotophytus, the subadult males develop the coloration as well. I was just curious if you considered testing the hypothesis that the coloration in the adult females actually, in addition to indicating that they're gravid, or maybe that it also has an aggression suppression feature in adult males, for the adult females as well. I basically suggested this possibility for crotophytus because subadult males only have coloration that looks like gravid coloration in females. And why would they have that if it doesn't benefit them in some way?
No, we didn't test that hypothesis. In this species, maybe it can reduce aggression, but I don't think, because the color that the females have when they are gravid is white. When they are trying to reject copulations, they have this white coloration. So the red
really,
color,
after they're already gravid, they lose the gravid color. So after copulation, they lose the gravid coloration immediately, or they carry it through to the point where they get.
no, during the mating season they are red, and when they become gravid, obviously gravid, then they are white. So they lose their red color.
Well, how quickly?
Well, not so quickly, in weeks.
Over the course of weeks.
Yeah, 1 month.
So I'm just I'll follow this up with one last little bit. So I mean, once they're gravid, I mean, where it no longer benefits them to be.
You mean at the beginning of being gravid?
Right. So now the males are still attempting to copulate with them, presumably.
Yeah.
And how do the females react to males in that instance? And how do males react to females? I mean, do you have a sense of that?
Not really, because observations in the field, we don't have observations in the field for these interactions between males and females. Usually, these males approach to females and they are aggressive, but the reaction of the females, we don't really know.
To what extent are females choosing? Is there variation among males that the females are cueing into?
Well, yes. we haven't studied that because the males don't have this red color, and so we were focused on the red color. But males have, for example, yellow spots on the sides, and of course there is variation in the expression in the number of spots and in the intensity of the yellow color. So probably they are choosing, but. Yeah, well, it's difficult to say. In reptiles, it's sometimes difficult to say if there is any choosing involved, because when a female is trying to copulate with a female, it always looks as a forced copulation. So probably some females can fight harder than others to avoid the copulation, but they don't know if they really choose anything.
So if you prevented the females from copulating, do they retain, and then they don't become gravid? Do they retain the red color?
They become gravid.
No, but let's say you don't let them mate. Will they
No,
retain the red?
well, we had in the last experiment, we had females that were not allowed to copulate with a male, and they became gravid, and they laid eggs.
Infertile.
Yeah, but they change color also.
Yes. In the same way as the others.
Yeah,
exactly the same. So it doesn't matter if their eggs are fertile or not.
Right.
So maybe I missed this at the beginning. Do they have any tail autonomy? Do they lose the tails at all when they're
Yes.
attacked? They do. Okay. Even as adults, that can happen.
Yes.
Okay.
Also bones.
So that anti-predator function might persist in stages beyond the.
Yeah, there is on one side there is the anti-predator function of losing the tail, but in addition to that, juveniles with the red color are attracting attention to the tail. And juveniles are more active than adults. Probably they need more this. Maybe they are going to be detected anyway by predators, because they need to be active, because they need to eat more or during more time to reach the certain size or to survive hibernation, for example. And then if I must be active and I will be detected anyway by predators, so maybe it's a better strategy to have this decoy. That's our interpretation. Yes,
So the males, they still court the females when they have white tails?
yes. they,
So it's just hard for me to imagine that red coloration is, I mean, it seems like it's such a risky thing to have that red coloration on the tail. Your experiments with the models show that the red tails get attacked more frequently.
yeah,
And so what's the benefit to a female to have that red coloration?
when this red color on the dorsal part of the tail is lost already when they are juveniles. It's only in hatchlings, when they are very small, when the red color is. So the red color is less visible to predators in juveniles and adults. In any case, yes, they are still more visible than having just other color. And the benefits for females of having, I don't know really. I have to think more about that.
I know I'm beating a dead horse here, but I'd really like to know if these females show the underside of their tail so the males as part of their rejection display. Chronophytids and other lizards have a special rejection display that they use when males are courting them when they don't want to mate with them, the sidle hopping thing, and they arch their tail and they show their sides. I'd be curious.
I haven't seen that behavior, but well, I can't say for sure that it doesn't exist. I haven't seen that.
Relating to hue intensity and detectability, it's kind of interesting to me that at least with my human visual spectrum that as hue intensity is increasing, the most detectable to me would be the intermediate values of hue because they're the brightest. Whereas when you get to a more intense hue, it actually appears more brown to me and looks more cryptic.
Well, in the hue.
There we are.
Yeah, but these high values of hue, in fact, they are here. They are in the red, and these other are in the orange. I'm not sure. Is
Yeah,
is
okay.
is red less conspicuous than orange in the field?
It depends on the substrate, I suppose.
I'm not sure. Maybe, I don't know, there are more orange things in the field where they live. Maybe leaves from the previous year, they are more common with orange coloration instead of red coloration. I don't know, I'm guessing.
Yeah, I hadn't noticed the difference in scale on the x-axis between them. Yeah.
Yeah, related to that, is there geographic variation in the red color throughout the range, or?
A little bit, but well, I only have 2 populations, so
Okay.
I cannot say. But in the south of Spain, they are redder than in central Spain, but with only 2 populations.
You have a question in the back there.
Okay, a couple questions. One, have you ever looked to see if the females are putting any of the pigments in their eggs? Or
If they are putting any?
if they're putting any of their pigments in the eggs?
I don't know. It's in other species. It's common that they put some carotenoids in the eggs, but here, well, the pigments responsible for the red color are pterins, not carotenoids. But there might be a relationship. Maybe, I don't know.
Yeah.
They are using carotenoids for the immune response instead of pterins, or the other way around. They have more of the other pigment to allocate in the eggs. I don't know. No, but I haven't studied that.
Yeah. Another question is, do any of the adult males have red tails?
Adult male red tail, no. No, none of them. They lose their red color when they are around 1 year old. And
So if a lizard with a red tail loses their tail and they grow back a tail, do they always grow back the same color, or do they?
No.
Is it white the second time?
yeah, it's not even the same white as the normal tail. It's kind of grayish.
And does that matter by life stage, like juvenile versus adult female?
I I haven't studied that systematically. Well, from observations, I think I don't think they regrow red tail.
Okay, well, if there are no more questions, thank you very much.
Thank you.